Okay, we're going to go ahead and get restarted now. So we now shift from disparities in cancer research to racial biases in the design of medical devices. And for that is our next speaker, Dr. Michael Sjoding. He's a clinical assistant professor of internal medicine in the Division of Pulmonary and Critical Care here at the University of Michigan. And he's also a member of our Center for Computational Medicine and Bioinformatics. Dr. Sjoding received his medical degree from Loyola in Chicago, followed by his internal medicine residency at the University of Chicago. With grants from both the NIH and Department of Defense, his research focuses on developing new computational tools to support diagnosis and to identify optimal treatment strategies for patients with acute respiratory diseases. He also studies the epidemiology, healthcare delivery, and outcomes of patients with acute respiratory distress syndrome, or ARDS. And the title of his talk will be Hiding in Plain Sight, Racial Bias in Medical Device Design and Its Impact on Patients. So I'll hand it over to you, Dr. Sjoding. Thank you. You're welcome to take your mic.
Yeah. Thank you. They can see me? Okay, great. So I'm going to change gears a little bit from what we've been talking about already today and focus really on medical devices. I think people appreciate how there can be bias in computational tools or software or algorithms, but I don't think people have as good of an appreciation of the fact that this can actually seep into medical devices themselves. So I'm going to talk about a few aspects of how racial bias can be introduced and perpetuated in medical devices. These include aspects of their physical design, some sort of physical property of the device that impacts patients differently based on their disadvantaged status. Talk about how data used in the development of the device or data that is generated by the device and interpreted differently can introduce racial bias. And finally, although this can't introduce bias, evaluation and regulatory standards are often quite insufficient to really appreciate how racial bias is present in these designs. I'm going to talk about two different medical devices that are critically important for my field of pulmonary and critical care. I'm going to introduce some of my work on uncovering racial bias in pulse oximetry. And then I also want to talk about spirometry. This is the measurement of lung function, which also has been shown to be biased. And I want to stop just for a second and mention one thing before I move on. There have been some sort of murmurs, can a medical technology be racially biased? Some people have sort of questioned that, a technology can't overtly make a decision to be less accurate in a certain group, so it can't be biased. But I'm going to take the argument put forward by a sociologist, Ruha Benjamin, in this really great book, Race After Technology. And she argues that racial bias doesn't require ill intent. So in healthcare, it's well described how physicians or other healthcare professionals can have unconscious or implicit bias that influences their medical decisions, and they don't even realize it. And so similarly, I argue that technologies or algorithms can be racially biased due to flawed design. And because of that flawed design, they can perform differently in specific groups. So the pulse oximeter turns out to be this really incredible technology. So what it allows you to do is to non-invasively measure the oxygen content of a patient's blood. And it does this by shining light in two particular wavelengths through the finger. And based on how much light is transmitted or absorbed, you can use some basic calculations to estimate how much oxygen is in the blood. And it uses two specific wavelengths because those wavelengths of light happen to absorb oxygenated hemoglobin and deoxygenated hemoglobin differently. And so based on how much of those two wavelengths of light get transmitted, you can sort of figure out what the oxygen content is in the blood. Further, when you actually send light through the finger, actually the vast majority of it does get absorbed, not by blood, but by other things like your bone, like your tissues, like your skin. And so just a small amount of light is actually absorbed by our arterial blood. And so when you sort of measure this absorbance over time, you get this wave called the photoplasmography, and that's what's depicted here. And so if you can isolate the peak and the trough absorbance of light during these cardiac cycles, you might be able to isolate the absorbance due to blood. And so then if you're taking the peak and the trough absorbance of light at both of these wavelengths, you get to this value called the ratio of ratio, called the R value. And this R value has a direct relationship with the actual oxygen content of the blood. And you can't actually go from the R value directly to oxygen content with this a mathematical relationship. You actually need to determine this relationship empirically, and it's largely linear, but you need to collect data on individuals who you're applying this device to sort of measure and come up with this calibration. And so there's a few assumptions in this device design, which ultimately is really quite magical. The first assumption is that by isolating this peak and this trough, you're really sort of taking out of the equation other aspects, such as the patient's skin thickness, the patient's skin pigment, and the finger size. And then sort of a related assumption is that this R value relationship with arterial oxygen is going to be constant, or is going to be the same across all patients. And it turns out both those assumptions turned out not to be particularly true. And so, after the pulse oximeter was introduced in medical practices in the mid-'80s, there were some studies evaluating the accuracy of the pulse oximeter in various patient populations. And so mid-2000, there was a small group who brought patients, healthy individuals, into a laboratory setting, and they measured oxygen levels by the pulse oximeter and the arterial blood gas, and they did find that there was some differences. And there seemed to be a bit of a dose-response relationship with skin pigment level and accuracy of the device. And they concluded in this small study that dark skin pigment decreases the accuracy of the pulse oximeter in low saturations. So they only had 36 healthy subjects to do this type of analysis, 17 darkly pigmented, 7 intermediate, and 12 lightly pigmented. A small study published in a second or third tier anesthesia journal, didn't really see a lot of light of day. Most people didn't really pay attention to it. Myself, I'm a trained pulmonary and critical care physician who uses a pulse oximeter every single day. I had no idea. Fast forward 20 years, and we're in this COVID-19 surge. And the first part of the surge, you probably all don't remember at this point anymore, but there was this huge surge in the greater Detroit area. And so in the first surge, COVID really disproportionately impacted minority groups like Black patients. And at the University of Michigan, we saw this huge spike in COVID cases in Black patients, and a much higher rate of patients hospitalized at the University of Michigan who were self-reported as Black relative to sort of normal times. And then in this context, turns out the pulse oximeter turns out to be incredibly important. It's sort of critical tool for caring for these patients. And fortunately, a woman, Amy Moran Thompson, who's a sociologist at MIT, sort of digs up some of these old studies, these sort of studies that no one really paid attention to, and sort of describes how, gosh, these studies could be influencing, these studies and these small differences in the pulse oximeter could actually be influencing outcomes of patients. And, fortunately, I saw this study, some of my colleagues studied this study, and we got incredibly concerned that, gosh, the pulse oximeter, which is this incredibly important medical device, could be impacting patient care in a way that could be very problematic for certain groups of Black patients. And so we set out to perform a study in a completely different way than it had been done before. So rather than taking patients into a lab to measure the pulse oximeter and the arterial oxygen content of their blood, we use routinely available electronic health records. We all, our electronic health records are now digitized. And so, the access to data is sort of incredible relative to what it was 10 or 15 years ago. And really similar to the approach taken in the previous study, this data was collected as part of routine care. So it really wasn't used or collected in a way that was sort of intentional to answer a question about is the pulse oximeter accurate? Nonetheless, what it provided us is 1 to 2 orders of magnitude more patients available to answer this question. And so just using University of Michigan data, looking at patients who were self-reported as white or Black, hospitalized between January and June of 2020, we were able to get pulse oximeter readings followed by arterial blood gas results on over 1,000 white patients, over 250 Black patients, and 10,000 total samples. And when we analyzed this data, we saw that there was this clear discrepancy. That here on the x-axis, this is what the pulse oximeter is reading. When the pulse oximeter is reading 92%, which is a percent of saturation we tend to think is going to be okay, the true arterial oxygen saturation in Black patients was actually meaningfully lower than in white patients in a way that likely could be problematic in terms of the care that they're provided. And so what we did was we asked this question, gosh, when the pulse oximeter is reading between 92 and 96%, how often is that true saturation less than 88, which is actually a problematic level, a level that could be causing harm, and a level, if I knew was actually happening, I would change how I would care for this patient. And so in white patients, this didn't occur that often. The pulse oximeter wasn't way off that often, 3% of the time. But in Black patients, it was 3 times as high. In a problematic way. And so we termed this sort of occurrence occult hypoxemia, when the pulse oximeter seems to be reading normal, yet in reality, the true arterial oxygen saturation is quite low and problematic. And we found that both in an analysis that was unadjusted and an analysis that was adjusted for other factors, such as a patient's age, sex, and their sort of cardiovascular organ dysfunction quantified by the SOFA score, that this difference persisted. We also did a few other analyses just to confirm that these findings seem to be sort of robust and hold true. So we looked at the pulse oximeter's ability to detect a low oxygen level of less than 88%, which everyone would consider is problematically low. And it turns out in Black patients, the area under the receiver operating characteristic curve to detect this level of oxygen was 0.84. Whereas in white patients, it was 0.89. Not huge difference, but think about how common the pulse oximeter is how often it's being used across hospitals all over the world. Small difference can really add up. And then, because again, these electronic health record data sources are, becoming more and more available, we were able to perform the same analysis in a different cohort in patients hospitalized and in this cohort called the Philips EICU cohort. This was released by the MIMIC data team. It's data from 178 hospitals. And so we were able to analyze 8,000 patients in 2014 and 15 and did the same analysis. How often was the pulse oximeter reading normally? But the true arterial oxygen saturation was low, 6% of the time in white patients and 17% in the Black patients. This is quite reassuring because, it wasn't this finding that we found wasn't sort of just a weird nuance of University of Michigan data. University of Michigan happens to use a particular brand of pulse oximeter. So it wasn't just this particular brand of pulse oximeter. It was pulse oximeters all over the country. And gosh, this finding has been hiding in plain sight. It was present in this Philips EICU cohort data from 2014 and 15, and we didn't even analyze it and look at it until 2020. So there was some limitations to this approach. And, I think that's been a theme that we've already heard twice already this morning, that, the types of data that you need to uncover these, to perform these analysis are messy. We chose deliberately to take a new approach rather than perform analysis in a laboratory setting with perfect data. We use the messy electronic health record data that's real world. And so one consequence of this is that the pulse oximeter values followed by the arterial blood gas values aren't being performed at the exact same time. And that's, after this study was published, that's the biggest irk, the biggest irk of these medical device companies. Like, how could we use this data to demonstrate this disparity that's not perfect? But, I think it's perfect, that it's good enough. If the pulse oximeter is being recorded followed by the arterial blood gas, and it's 4 minutes later for both black and white patients, gosh, like that just wouldn't explain why this disparity exists. The other limitation here is the analysis was based on self-reported race, which correlates with skin pigment, but within racial groups. Obviously, there's differences in skin pigment. And finally, we were unable to quantify the clinical impact of these differences. But since our study that was published last year, I think there's been 5 or 6 follow-up studies confirming these findings, and 2 recent studies showing that this disparity in the pulse oximeter's ability to detect low oxygen levels have impacted outcomes. And so, thankfully, after our study was published, the FDA did issue a safety communication saying that there are lots of things that can impact the pulse oximeter's accuracy. Factors such as poor circulation, skin pigment, skin thickness, temperature, tobacco use, fingernail polish. And they wanted to remind healthcare professionals that this device is imperfect. And they wanted to remind health professionals to consider these limitations when you're making treatment decisions, like who should be admitted to the hospital, who should get particular treatments. But what the FDA has not done yet is actually this last issue in sort of the three things that can cause and perpetuate racial bias in devices. And that is, they haven't changed their guidance for how these devices get evaluated in the first place. So if you were a medical device company and you had a new pulse oximeter that you wanted to bring to market, you have to provide data to the FDA saying that your device is effective. And to do that, you need to show data of 10 or more healthy subjects that vary in age or gender and 200 or more data points. And at least 2 darkly pigmented patients, or 15%, should be in your sample. And that right now is the extent of the FDA's regulation of pulse oximeters. And anyone with any sort of knowledge of data would appreciate the fact that having 2 darkly pigmented subjects in your sample is not going to be enough to really be able to discern whether there's actual real bias in the pulse oximeter based on things like your skin tone. So in summary, how was racial bias introduced and perpetuated in pulse oximeters? There were issues with the physical device design. There was assumptions that, when you isolate the peak and trough of the wavelengths of the cardiac cycle, you can get away from having influence on things like skin pigment. The development data was problematic because it's likely the case that when pulse oximeters were first developed, that they largely used white male patients to sort of calibrate these devices. So of course they work better on white male patients than on other patients. And then the evaluation and regulatory standards were insufficient. I want to also just mention this other medical device that's fundamentally important to providing pulmonary care, because I think it's a nice complement to the discussion about pulse oximeters, and that's pulmonary function testing. Pulmonary function testing is used to diagnose respiratory diseases and make treatment decisions. And when you do population health studies measuring patients' lung function, what you find is it's often the case, and this has been shown over and over again, that lung function is different across racial groups. and patients who self-report as African Americans or Mexican Americans typically over their lifespan have lower lung function than white patients. And so when you're developing a technology like a lung function test and you want to provide useful information to a physician, you know, when a patient performs this test and gets a measurement like an FEV1, which is a measure of lung function of 4 liters, gosh, how do you appreciate or understand whether that's normal or not? Well, so what you do is you create ranges of what normal equal. And so you come up with an equation that tells you, you know, what's normal. And then you apply that equation so that you know in your clinic that this patient's lung size of 4 liters turns out to be normal. And unfortunately, back in when these lung function testing were becoming more and more prominent, investigators decided that we needed race-specific lung function equations of normal. And that was actually based on some really problematic science that suspected that there was actually anthropomorphic differences between white and Black patients, in that Black patients had lower lung size. And that was the justification for creating race-specific normals. So here, you know, in contrast to the pulse oximeter, the lung function test, you know, there's nothing sort of racially biased about how the test is performed, how the lung function is measured. However, instead, in this case, the racial bias occurs in how you interpret the results. And, you know, thankfully, there has been appreciation of the problematic nature of these decisions to generate race corrections or race-specific lung function. And there was just recent study published on this issue. This is a remarkable study. What the investigators did was they took a large population of patients with COPD, and they looked at these race-specific lung function equations and how they align with other things that measured respiratory health. So here I'm showing the lung function and measures of respiratory health as quantified by respiratory symptoms. And so on the right side here is the race-specific lung function equation. So if you look on the x-axis at 100%, 100% means normal, 100% means good, healthy lung function. And then if you look at these two regression lines, what you see is that Black patients at supposedly quote unquote normal lung function have a higher degree of respiratory symptoms of cough and shortness of breath. So, I mean, for me as a pulmonologist, this is really scary stuff. What that means is a patient who actually thinks they have a real problem, they have real respiratory symptoms reflective of a real disease, could come into clinic and then be seen by a lung physician, and lo and behold, because they use these race-specific lung function equations, they have supposedly normal lung function and they're told they're normal. But when you actually not use the race-specific lung function equation, when you use the universal lung function equation, you see that that gap disappears. Now that people with the same level of respiratory symptoms like cough and shortness of breath also have the same level of lung function. And you know, one thing you can see about this data is it's awfully messy, right? Like that's the story here that we've been telling a lot today. It's messy data, but despite that, the findings are quite clear. The other thing that they did in this study that I think is just really cool is they used quantitative evaluation of CAT scans. So CT scans of the lungs. Turns out we have this amazing ability to do quantification of lungs. And one thing that you can do is measure the size of the airways and the thickness of the airways. And patients with thicker airways have more disease. And they looked at the association between the thickness of the airways and these race-specific and general lung function. And lo and behold, when you use race-specific lung function equations, it makes it seem like Black patients with normal lung function have thicker airways. That doesn't make sense because that would suggest that they're more diseased. But when you actually use the general lung function equations, it better aligns with this quantitative measure. So, you know, in the case of spirometry, really the problem was in the data interpretation, and there was some issues with the evaluation and the regulatory standard of spirometry. So I want to just close right now, and then I'm happy to take a few questions. I think sort of hopefully one theme across my work and others who are working in this space is that using new data sources and analytic approaches really are keys to uncovering potential bias in medical devices. For our work, it was using a different data source, electronic health records. For the pulmonary function testing work, it was using new data sources like respiratory health questionnaires and quantitative CT scanning, and sort of new ideas, breathing new life into these fields can help uncover these biases. And I just want to describe here how bias can be presented in both hardware and software. I think people do have a general sense that algorithms can be biased based on the data that they're trained, but the roots of these biases can be deeper and really can be in the hardware of the devices themselves. And understanding how these biases originate and perpetuate are essential to developing robust safeguards for medical devices. So I just want to also conclude, if you're interested in this area, there's two really great articles that I would recommend you reading. The first is Hidden in Plain Sight, Reconsidering the Use of Race Correction in Clinical Algorithms, which is an article published in New England Journal of Medicine 2020. And then an article published in Science, really sort of focusing on our work and then about pulse oximeter and then work more generally in ensuring medical devices are fair, was published in Science in 2021. So thank you very much. Sure. All right.
Is this on? Okay. I'll ask one question. So where do you see the oximeter device going in the future? Like, I would think they could make a device that could automatically detect the pigment in the skin and then adjust the function based on that. But I guess it's not clear to me like how important these other things are, like the skin thickness, and if that is correlated with pigment or other things like that.
Yeah, great question. So, in talking with engineers who are expert in this area, like you said, like the engineering solutions aren't like groundbreaking. We don't need to do some sort of like discovery of new engineering technologies. So yeah, just like a simple device on the pulse oximeter that actually quantifies skin pigment level could recalibrate. Other work has described how, if you can use multiple wavelengths rather than just two, you can get a better estimation. So I think the research today probably shows that actually that's not even true. So we have recent work that's about to be published showing that pulse oximeters work less well on kids, and kids have smaller fingers. And so it turns out, like, as the pulse oximeter is sending light through the finger, smaller finger, more scatter. So yeah, I mean, I think there does ultimately need to be a more complex design change for this to be effective. My sense is it's going to require a few different approaches, one of which is using more wavelengths of light to derive more better calibration. Doesn't look like it.
Okay.
Thank you.
Yeah. So thank you, Dr. Sjoding. Do you want to